Five friends spectacular. Five friends spectacular. Five friends spectacular. Five friends spectacular.
The mitochondria is the powerhouse of the cell.
Five friends spectacular. Five friends spectacular. Five friends spectacular.
Sounds so horrible.
You are about to embark on a journey through space and time that will change your life as you know it. Or should I say, your bio as you know it. Our journey will encompass the beginning of life on Earth and end with examining your body today.
My body?
Yes, your body.
That's creepy.
Follow me as we turn back the clock billions of years ago to the harsh climate that was the Earth. Water was only in the form of vapor and ice, and volcanoes exploded everywhere. This harsh environment created the first organic molecules, and life just appeared randomly out of thin air. Dinosaurs started climbing out of the earth, and some DNA was brought down on like a meteor, and then everything started.
Obviously, that's not even right.
Yes, of course I was joking. Could you not tell by my hysterical laughter? Do you know what makes a molecule organic?
Carbon, obviously.
Yes, carbon. The first organic molecules were synthesized through the carbon in the atmosphere, and eventually genetic material arose. The first prokaryotic cells relied on this genetic material called DNA. All cells rely on a specific class of molecules called macromolecules. There are 4 different macromolecules. The first one that we will talk about is protein, bruh. Now, everybody knows that proteins are used to build muscles and, of course, get swole, but proteins actually have many more functions than just building muscle, such as acting as enzymes, hormones, and structures. Amino acids are the monomers of proteins, and these bond together to form polypeptide chains. You can tell a molecule is an amino acid by the presence of a nitrogen atom. There are 20 different amino acids that humans need. 8 of them are produced by the body, and the other 12 we have to ingest through our diets. Proteins are synthesized in ribosomes, and DNA codes for each specific protein. There are also lipids, or fats. Fats are essential to life, but most people just think that fats are the things that hang off your love handles and you want to get rid of. But in fact, fats are essential, and you would die. Fats cushion your organs, help you retain body heat, are used as hormones in your body, and are used as a way to store energy in your body. Phospholipids are also an important part of the cell membrane. Fats can either be saturated or unsaturated. Unsaturated fats are things like olive oil and any other nut oils out there, and saturated fats are found in animal fat, such as in butter and like steaks and stuff, all the good fat, saturated fat. Saturated fats are solid at room temperature because the molecule is straight due to being completely saturated with hydrogen molecules. Unsaturated fats are in a bent shape, unlike saturated fats, because the carbons are not fully saturated with hydrogen bonds and instead form a double bond between themselves. Another part of the mosaic of the cell membrane is the carbohydrates. Carbohydrates are our 3rd macromolecule, and they come in a 1 to 2 to 1 ratio of carbon to hydrogens to oxygens. Most heterotrophs rely on carbs as their main source of energy. The monomers for carbohydrates are glucose, fructose, and galactose. Glucose and glucose form to make maltose, glucose and fructose form to make sucrose, and glucose and galactose form to make lactose. There are also a few polysaccharides as well. For example, starch stores energy in plants, and glycogen stores energy in animals. Polysaccharides can be thousands of monomers long. All life is nucleic acid. The rest is just commentary. Quote, kid from the yearbook. We finally have our nucleic acids, RNA and DNA, deoxyribonucleic acid and ribonucleic acid. They code for all the hereditary information in the universe. Nucleic acids are made out of monomers called nucleotides. Nucleotides are made out of a phosphate, a 5-carbon sugar, and a nitrogen base. In DNA, the nucleotides are adenine, guanine, cytosine, and thymine. But in RNA, the thymine is booted out and replaced by uracil. Adenine mixes with thymine or uracil, and guanine and cytosine pair. Stay tuned to see more after this commercial break.
Gardening is hard work. You've heard of fertilizer, you've heard of manure to help your plants, but have you heard of Sprite? Nope, not for drinking. You pour it on your plants and they will survive and thrive.
Sprite. Get yours today.
And we're back.
Hello, my name is Dr. Doofenmertz. Right now I'm in my lab here in Siberia. So happy all of you could join me here today. Now because we're in a lab, safety first. Alright, now we can begin. So before we dive into all of that fun stuff you kids were talking about, we have something special in store for you. And that is a chemistry lesson. This is crazy, I know kids. But just how bio is life, chemistry makes up those elements of life. First, everything in this world is made up of things called atoms, and I know you've heard of these because they're all around us. Now let's take a look at the periodic table. It says every single atom in the entire world on it. There are things such as chlorine, which you'll see in a pool when you go swimming in the hot summer, or potassium, which you'll find in your favorite snack, a healthy banana. Now within each of these atoms, there are even smaller pieces. There are protons, which I'll use blue to represent, and these have a positive charge. So to remember that, you can think proton with a P equals positive. Now there are also electrons, which I'll use red to represent. They have a negative charge. And lastly, there are small things called neutrons, which don't have any charge. And I'll use green to represent those. So the little protons just like to sit right in the middle of the atom very comfortably. And with them, the neutrons are their little buddies, and they like to sit with them too. So if there are 5 protons, there should be 5 neutrons as well. But here's where the family drama gets a little bit saucy. That's when the electrons come in. These guys are energetic and the enemies of the protons. They like to fly around the edge of the atom, and they move all around, so they're not stationary at all. But together, they make up an atom. Now everything in this world, such as this glass here, or even this marker, aren't just made up of one type of atom. They're made up of many that come together and form what's called a compound. Now to understand compounds, let's take a look at this average car right here. So hello again. So like those compounds I was talking about, a car has many components that are necessary for it to work. So we have the wheel, so we have our key to start the car, and we have an engine so that we can drive. And for safety, we have a horn, and we got windows. We also have the brindle. Thanks for joining me on our little field trip outside. But now that we're back in the lab, let's get down to some serious science. Let's talk about compounds using salt. Now I'm gonna let you in on a little secret. Salt isn't this guy's real name. It's actually sodium chloride. Now what does that mean? It means every little grain is made up of a negatively charged sodium atom, as well as a positively charged chlorine atom. Chlorine has 7 electrons in its outer shell, just floating around happily, while sodium has just 1 electron. These guys form an ionic bond together when they make salt. So sodium gives one of its electrons to chloride, and they are forever in a happy bond. Compounds can be made, though, from more than just ionic bonds. There's also something called covalent bonds, which what your favorite sugary snack has inside of it. So sugar is made up of carbon, oxygen, and hydrogen. And all of them just share their little outer electrons together in one happy, sharing is caring family. So that concludes our show for today.
Yay! Chemistry's over! Woo! Get back to bio!
That's right, kids. You made it through your first chemistry lesson ever. Thank you for joining me, Dr. Dathan Sertz.
Welcome to the new segment of our show, Cookies Are Us, brought to you by... Today we'll be making lots of dividing cookies. Let's start with mitosis. In the S phase of interphase, the DNA is replicated. In prophase, chromosomes condense and the nuclear envelope starts to disappear. During prometaphase, kinetochores appear at centrosomes and mitotic spindle begins to form. During metaphase, the chromosomes line up at the metaphase plate, and in anaphase, sister chromatids are pulled towards opposite poles. In telophase, a cleavage furrow begins to form and splits the cells during cytokinesis. The nucleuses reform in the two new identical daughter cells. Again, here's mitosis. Thank you for watching this Cookies R Us bio video. Join us next week for another cooking dividing adventure.
Stay tuned. We'll be right back after this commercial break.
I used to think I was just from Earth. But then with the help of Mendelian Incorporated, I found out I was from out of this world! Now you've heard of Ancestry.com and you've heard of 23andMe, but you've never heard of Mendelian Incorporated before. Utilizing 19th century techniques from the father of genetics, Gregor Mendel, you can find out exactly where you're from in just 4 easy steps. Step 1, order a kit with everything you need in it for just 17 payments of $49.99, and the kit will be shipped straight to your door within 5 weeks. Step 2, now that your kit's been delivered, just open it right up. Now you have 2 options for sending us your awesome DNA. Step 1, take one of the included swabs and swab the inside of your cheek. Like so. Or step 2, if you're still youthful like I am, you can take some of your hair. Step 3, take your DNA sample, again either that swab or your youthful hair, and put it back into a sterile bag, again included in your kit for that one low cost. Just seal it up. Like so. Step 4, now all you have to do is send the kit back, and in 2 to 28 weeks your results will be in. Don't be lied to any longer. So order now! Order within the next 15 minutes and you'll get free processing and handling off your entire order. But that's not all. You'll also get a free can of Sprite. Ah, refreshing. The time is ticking. You'll never find a deal that's quite so out of this world.
And we're back!
Hey everybody, who's ready for another cookie? Just kidding. Coffee filter dividing adventure! First we have interphase, and in the S phase of interphase, chromosomes are duplicated and the sister chromatids are held together by centromeres. In meiosis 1, we're going to start off today with prophase 1. In prophase 1, chromosomes condense, binding together, forming a tetrad. Prometaphase 1 is up next. Spindle fibers extending from the centromeres begin to form. Then, in metaphase 1, homologous chromosomes assemble at the metaphase plate from held by the microtubules. Anaphase 1 is next, where the microtubules pull the homologous chromosomes apart. In telophase 1, a cleavage furrow begins to form, and in cytokinesis, the cell separates into 2 separate identical daughter cells. Meiosis 2 now begins, starting with prophase 2, where sister chromatids condense and a new spindle begins to form. In prometaphase 2, the nuclear envelope disappears and the kinetochores attach to the centromeres. In metaphase 2, sister chromatids line up at the metaphase plate. Anaphase 2 is next, where the sister chromatids are pulled apart by the kinetochores. Through telekinesis 2, a cleavage furrow divides each cell into 2 cytokinesis, creating 4 haploid identical daughter cells.
Now let's speed up the process.
And that's a wrap, folks. Catch us next time on another cell dividing adventure.
Hey doctor, can you tell me why I act so crazy around that hot boy from school? Now that's a good question. So kids, what gives us the feels? I know, the heart! No. As many of you probably already know, blood in humans flows through a closed circulatory system, through veins, arteries, and capillaries. The heart creates a pressure that allows the blood to flow throughout the entire body. It is powered by a cardiac muscle, meaning if it were taken out of you, it would continue to contract and relax. Anyone else have any ideas on what makes you so nervous around those hot people? Is it the nervous system? Nope. That's responsible for receiving, sending, and processing nerve impulses. Neurons send electrical impulses to your central nervous system, where the message either gets sent to your brain to make a decision, or sends it directly back with a response saying, "You're stupid! Get your hand off that burning stove!" It's the hormones and the endocrine system. Come on, guys. There are two types of hormones, lipid or steroid hormones and protein or peptide hormones. Lipid and steroid hormones are hydrophobic and can diffuse through the membrane. They bind to receptors inside the nucleus. An example is testosterone, your favorite. It is secreted by the testes and acts as transcription factors. Only certain cells have their receptors inside their nucleus, so this creates a concentrated response. That way, when the hormone is secreted, not all your cells are rapidly growing mountains of hair everywhere. The most common protein hormone is epinephrine. It is responsible for the flight or fight response. It cannot diffuse through the membrane, so it binds to receptors on the cell surfaces and signals a cascade effect. It signals for the liver to release glucose, convert glycogen to glucose, and inhibit glycogen production. This is very useful for when that random lizard comes out of nowhere and starts attacking you. So now that you know why you act so crazy around that hot boy, just know that it happens to everyone. So you just gotta embrace the crazy, woo!
Stay tuned, we'll be right back after this commercial break.
Do you ever find yourself finding it very difficult to complete simple tasks, such as drinking soda?
What are you doing? Drink water! It has a high specific heat! It moderates temperature! It's adhesive and cohesive! And it's used for hydrolysis! Ice floats. How is that relevant? You die without it.
Water. Drink yours today. Found in your local supermarket and probably inside your house. May be carrying diseases and not responsible for any injuries. If there becomes a hypertonic solution, the cells may shrivel, and if it's a hypotonic solution, your cells may less. Supplies are limited. Excess water may cause stress on heart and kidneys and can even lead to death. Not responsible for any deaths. Thank you. Enjoy your water.
Hello, living human beings. Today, we're going to be making something that's not living, mostly because it's made out of paper and pipe cleaners, but also because it's a virus!
A virus is a non-living thing that injects its DNA into a host cell in order to reproduce.
Bam! Look what we're going to be making today. It's called a bacteriophage. Why is that? Well, bacteriophages only put their DNA into bacteria.
If you'd like to make this beautiful model with us today, please head to www.coolestbioshowever.com and follow the links below to find out a printout that looks just like this. But kids, be careful. You must be 18 years or older in order to go onto this website, as well as use scissors, which will be needed in this project. Step 1. Gather all of your supplies. For this project, you'll need our paper cutout. You'll need a pair of scissors. Please be careful with them. And then you'll need tape or a glue stick, your choice. We prefer tape here, it's a little less messy. And pipe cleaners. Boop, boop.
Step number 2, cut everything.
So currently, Charlotte here is cutting out some of the major components of a virus. But let's take a second to go through what each part is. So at the top here, we have the head, or the protein coat. And inside is all the little DNA that the virus uses to reproduce, again with the help of a host cell. Right here, we have what's called the neck, or the sheet. And down here, you have all of the different tail fibers. And the virus uses the tail fibers to inject itself into a host cell. So let's pretend that this is a host cell. A virus will come in and plant itself down right on top, and it will inject all its DNA into it. Eventually, the DNA will stay inside this new host cell and start to reproduce. And after a burst, a lot more DNA, a lot more viruses come out of the host cell. to perpetuate the life cycle of the virus. Step 3. Take all of your newly cut out pieces and fold along all of the dotted lines.
All of the dotted lines.
All of the dotted
All
lines.
of the
Not some, all.
Alright, step number 4. You're going to take some tape, or glue if you're fancy, and tape along the little tabs that say glue. Alright, so what I'm making right here is part of the protein coat, or head of the bacteriophage. This is where all the DNA of the virus is kept.
What I made right here is the sheath or the neck. This connects the head of the virus with all of the tail fibers, which we'll make in our next step. And if this part gets a little complicated, please ask a parent or a trusted older sibling or friend for help.
Another good thing to do is positive reinforcement. Sarah, you're doing great.
Thanks, Charlotte. Now I know I can do it. Please put a piece of tape right here.
Alright.
There we go. Look at that. Completed head and sheath all in one.
There you go.
So now we need to connect these together.
Alright, just tape on the little tabs.
So these don't say glue here, but you'll see them.
You're gonna glue them.
Use tape conservatively. Alright, it looks like we're all done.
But, one more step.
Step 5. Take out your pipe cleaners.
6, to be exact.
But if you have pipe cleaners that are too long, take 3 of them and cut them in half so they're about 5 inches long each. Now what we're going to do is we're going to make one fold about 1 to 1.5 inches into the pipe cleaner. Then we're going to fold this remaining long piece in half like so in the other direction.
So, do this for every single one of your pipe cleaners.
And again, these are the tail fibers of the virus.
Alright, when you are all done with folding your tail fibers, you place the smaller bended end into the sheath here, like so. Oh,
I'll help you out. You got this.
oh, thank you.
So yeah, just hold it like this so they just fall nicely into place. And then you take a fairly long piece of tape
Got that right here.
and squeeze the sheath down. Alright, thank you. And you're just going to wrap the piece of tape really tightly around the bottom here. Now don't worry if your tails move around because you can move them back later.
Make sure it's all really tight.
Yep, so nothing falls apart. Now we're just going to readjust where these tails are again. And flip it over.
There's your bacteria
Look at that!
Wow.
Good job, guys. You did great.
Excellent work.
Now, I hope you enjoyed joining us on the segment today. So be sure to tune in next week for yet another great craft. Thanks, guys. That was so hard. I need some positive reinforcement for making this. But high five!
Here on the show today, we're here to talk about flowers and the properties of water. So, as you know, flowers get water from the ground and they do this through their roots and it goes all the way up to the top. Even to the tallest tree, water is transported. And how does this happen? Through the properties of cohesion and adhesion. These are properties of water. And so, water travels through xylem and phloem, and this is possible through cohesion, which is the attraction between water molecules to themselves, which makes them stick together. And adhesion, which is the attraction to other surfaces. This allows for a pulling method up against gravity. So this is how, plants get their water. And to demonstrate this today, we have these white roses here. We're going to put some dye into them, and then you'll see how they change color. This is because it's coming from all the way down here, all the way up through the xylem and phloem, up to the leaves, and you'll be able to tell. So next thing that we're going to do with water to show another one of its properties is to demonstrate its hydrophilic and hydrophobic tendencies with different substances. So hydrophilic substances, for example, like salt, mix easily with water, unlike this, which is oil, which is hydrophobic. So when we combine these together, they're going to separate. First, let's demonstrate salt. It's pretty easy. Just dump a little bit in. And as you can see, when you mix it around, it'll combine pretty easily, no problems. But when you combine a hydrophobic substance with water, no matter how much you stir it, it's always going to rise to the top. Here, so you can see it more. So all of this is the oil, and all of this is the water and salt.
What is that mysterious ticking noise? It's not over here, and it's not over there. Small bacteria. Small bacteria.
DNA!
Small bacteria.
DNA!
Small
Plants
bacteria.
DNA!
Small
Plants
bacteria.
DNA!
Small
Plants
bacteria.
DNA!
Small
Plants
bacteria.
DNA!
Small
Animalia!
bacteria.
Animalia!
Plants
Animalia!
DNA!
Animalia!
Small
Animalia!
DNA!
Animalia!
Small bacteria.
Animalia!
Bacteria. Animals. Bacteria. Animals. Bacteria. Animals. Bacteria. Animals. Bacteria. Animals.
DNA!
I found the source of the ticking noise. It's a cricket. Yay! Kingdom Animalia.
You're still here? It's summer. Go! Go, go!